Hi and welcome everyone. I am going to deal with course code BOC 109, course title Molecular Biology and Genetic Engineering. The title of the unit is The Structure of DNA and RNA as Genetic Material. Name of the module, Structure of Nuclear DNA versus Organelle DNA. In this module, the outlines which you are going to study is structure of nuclear DNA, structure of organelle DNA, that is mitochondrial as well as chloroplast DNA, differences between nuclear DNA and organelle DNA. At the end of the module, you should be able to understand the structure of nuclear DNA, to analyze the structure of organelle DNA, mitochondrial DNA and chloroplast DNA, to summarize the differences between nuclear DNA and organelle DNA. Nuclear DNA. Let us start with nuclear DNA. We know nuclear DNA is a nucleic acid which is a polymeric biomolecule or a biopolymer found in the nucleus of eukaryotic cells. You can see in this picture, you can see a cell with a nucleus and the hereditary material, and directly a chromosome is shown, and the chromosomes is, you can see the double-stranded DNA inside that. Now, this structure of the nuclear DNA was given by Crick and Watson in 1953. Each strand is a long polymer chain made up of nucleotides. Now, you can see in this figure, in this picture, you can see the nucleus and the chromatin material condensed. You can see all these things, nuclear pores, just to show you the position of nuclear DNA. Each nucleotide in a DNA is composed of 5-carbon sugar, a phosphate group and an organic base. You can see here a 5-carbon sugar, a phosphate group and a nitrogenous base here. They are linked together. The two strands are made up of nucleotides. They are linked together by hydrogen bonds, okay, between the nitrogenous bases. There are purines, which are larger bases, which include adenine and guanine. Pyrimidines, which are smaller bases, which include thiamine and cytosine. Okay, all the nitrogenous bases are linked by hydrogen bonds, but the number of bonds differ. You can see 2 bonds here and 3 bonds here. Now, the bonding is determined by Chargaff's rule, which states that adenine will always pair with thiamine and guanine will always pair with cytosine. The phosphate groups are held together by phosphodiester bonds, and the bases are held together by hydrogen bonds. Now, we can see organelle DNAs here. As we have just seen nuclear DNA, let us see organelle DNAs. Organelle DNAs are present in mitochondria and chloroplast of the cells. You can see the mitochondria here. You can see the DNA present here. You can see the DNA of the chloroplast here, present in the stroma. Now, their presence, the DNA present in the organelle, is governed by endosymbiotic theory, which states that mitochondria and chloroplast were primitive cells without nuclei. Means they were prokaryotes that got inside of primitive cells with nuclei. They got inside the eukaryotes. That's the endosymbiotic theory. They came with their own DNA, and now they have their own DNA that code for their own proteins. Thus, mitochondria and chloroplasts are endosymbiotic in origin. Let's discuss about mitochondria. You can see in the picture, you can see the same picture here. You have all the parts labeled here. You can see the DNA of mitochondria here. Originated from the Greek word, mito means thread and chondrion means granules. They are cytoplasmic organelles, all of higher organisms, and they are also called as powerhouses of the cells. Carl Benda, 1898, coined the term mitochondria. Mitochondrial matrix has circular empty DNA. We use the abbreviation empty DNA for mitochondrial DNA. It was discovered by Margit Nass and Sylvain Nass. Empty DNA is inherited from the mother and therefore termed as uniparental inheritance. What is the structure and composition of empty DNA, that is mitochondrial DNA? It is a long chain of nucleotides composed of sugar, nitrogenous bases and phosphate group, just like the nuclear DNA. The level of packing is also similar to nuclear DNA, and it also contains histones. Empty DNA has only one chromosome that is in the form of circular genome of prokaryotes and is much shorter and codes for specific proteins used in the metabolism. You can see the mitochondrial DNA here and its different parts, different regions. Okay. Going to human mitochondrial DNA as an example for you just to see. The human mitochondrial DNA is circular DNA molecule with 16 kilobases. It encodes 37 genes, 13 for subunits of respiratory complexes and 22 for mitochondrial transfer RNA and 2 for ribosomal RNA. One mitochondrion contains at least 10 copies of its DNA. The two strands of human empty DNA are distinguished as heavy strands and a light strand. You can see the heavy strand and this side is the lighter strand. The chloroplast. You can see chloroplast here, the structure. In the stroma, there is distribution of the DNA. Chloroplasts are organelles in plants, all green plants and even algal cells. They have a circular chromosome composed of DNA. Chloroplast DNA, also designated as cpDNA, has genes necessary for proper functioning of photosynthesis and is useful in studying evolution. Young chloroplasts contain around 100 copies of DNA. You can see the chloroplast here as an organelle. You can see the stroma region, you can see the grana region, and you can see this cpDNA here, which is highlighted. Now, structure of chloroplast DNA, it has got this circular structure. Small single copy region is this side, long single copy region is this side, and it is separated by the other regions. cpDNA are circular and has 120,000 or 170,000 base pairs long. Okay, they are packed into nucleoids which has cpDNA rings. Nucleoids are dispersed in the stroma region. Many cpDNAs has two inverted repeats which separate LSC and SSC. See, these are the repeats which separate this small single copy region and long single copy region, which is there in the chloroplast DNA. Structure. cpDNA of higher plants has around 120 genes. The length of inverted repeats varies from 4,000 to 25,000 base pairs. It contains 3 ribosomal or ribosomal RNAs and 2 transfer RNA genes. To sum up, we have the differences between nuclear and organelle DNA. One of the organelle DNAs I will take to differentiate the two. Now, nuclear DNA is located within the nucleus of eukaryotes, whereas this organelle DNA, if you see empty DNA, is located in the mitochondria. There are 2 copies per cell. This contains around 100 to 1000 copies per cell. The structure is linear with open ends. The structure is usually closed with circular structures. Nuclear DNA is diploid, inherited from both parents, whereas organelle DNA, especially empty DNA, is haploid and coming from only one parent, that is mother, and therefore we also call it as uniparental inheritance, I mentioned before. The mutation rate is less in nuclear DNA, it's about 0.3%, whereas the mutation rate is generally higher in the organelle DNAs. These are some of the references which you can refer for the topic. Hope you have benefited from this module. Thank you.